today's topic is lipids. So let's get started without delay the time. So here we mentioned two type lipids: saturated solid at room temperature, it means high temperature, and they have a low entropy, and low kinetic energy, high enthalpy. Unsaturated liquid in a room and high temperature contain double bond, so they known as cis position, and saturated not contain double bond, so they're known as a trans position. So in unsaturated contain a double bond is known as cis position. Their double bond give kick to the structure and instability at the room temperature. So sometimes they are liquid at ambitious environment and low potential energy, high kinetic energy, high randomization, enthalpy low, entropy high. So here I mentioned a structure. It is a stirred form because they have no double bond, so it's transformed, saturated, highly linked, ordered, closed bank. But on the other hand, it's unsaturated, they have a double bond, so it's create a unstable form known as a cis. Okay, here we mentioned carbon trans and carbon cis form. Okay, so there's the types of the lipids: alpha alanine, M for so here L for the lauric, their melting temperature is 44 degree, and second one is 14. It's known as the maristic. Their melting temperature is 54. Third one is the palmatic, contain number of 16 carbon, and their melting temperature is 64. And fourth one is stearic. Their melting temperature is 69, contain 18 carbon long chain. And the second one is the arachnoid. Its melting temperature is 76, and it contains 20 carbons chain due to less because high kinetic energy, so not require outer temperature. Here you mention in all these three contain a 10 degree differentiation in temperatures, but after that when the number of carbon is increases, the number of temperature, the degree of temperature is reduces. Why? Because due to the less, because high kinetic energy, so not require outer temperature, its own energy is quite stable, okay, to melting it. Okay, because due to the high kinetic energy, low potential energy, high enthalpy, and we know that kinetic energy and the entropy is directly proportional to the temperature. 2 carbon high, it means 10 degree temperature. This rule follow in that area. Okay, lauric, maristic, palmatic, stearic, organic. Okay, here we mentioned PUFA is the double bond between C3 and C4, especially important in the human nutrient. PUFA contains double bond near methyl group. It is important in human nutrient that to the carboxy, and the carbon of the methyl group, that is carbon more distant from the carboxy group, is called omega. C3 and C, if a double bond contains in between C3 and C4, it's known omega 3. If a double bond contains C6 in between C6 and C7, it's called omega 7. Human require but not self-synthesize omega 3. Okay. These stuffs are not here. We also discussed about that in previous one. The fatty acid are conjugated to glycerol and sphingolipins of glycerol at both ends. After even conjugation, fatty acid retain their property. Glycerol in the tri-alcohol storage lipid होता है. It's a storage lipid. Ester formation, membrane lipid 1, and now fatty acid at third carbon at phosphate position. If this is the structure of the lipids, and now here we changes the substitute, and there also changes the lipids name. Okay, if here add serina is all that structure, add ethyne, the phosphatic additive. If is at the position of H, add serine, add choline, add ethylamine, add glycerol. According to the melting temperature, the unsaturated have a low melting temperature and saturated have a highly melting temperature. In fast food have a high lipid, that's why its aroma come after very long distance. So oh, कहीं पका बन रहा है ना. Prostaglandin, mucin, arginoma plus coxa. Okay, because mucin is a coating layer in our internal organs. Outside it protect our internal organ by the hydrochloric acid, so it is synthesized by the arachidoma plus coxa. And if you take non-steroidal anti-inflammatory drugs like arsine, they inhibit the arachidoma plus coxa, so it also prevent the production of the mucin, and it cause harmful because directly or indirectly, because if it is high concentration, so no mucin form and our internal organs are damaged by the hydrochloric acid. Okay, glycogen use oxygen form, arachidonic acid, phosphoglycerol. Okay, now install for polar, so DAG membrane bounded and install hand in cytoplasm. Here we look like a structure. Now its H position is changed and add phosphoinositol during signaling. Okay, install polar, so DAG membrane bounded and install hand in cytoplasm. Okay, third carbon of inositol by phosphoinositol inositol, third carbon, and cell cycle start and phosphate removed by P10 B. Okay, it's a part of the signaling, so please skip it. DAG membrane included PI3 phosphate, phosphate 1,3,4 triphosphate. DAG is inside the membrane. It's also part of the signaling, so it's also covered later on. Sphingolipid derived from the palmitic acid and serine, one arm 6C16, second and not phosphodiester, not ester binding type, C double bond, third arm not phosphorylated. And sphingolipid involved in the brain development and surface antigen of blood O, blood group O, blood group B, blood group A. Fatty acid tail is amide group in C2 position. Here 5 majors are some. Sphingolipid only one fatty acid. This is the common chain containing glucose, galactose, galactone, and style and galactose. If it contains the last chain hanging with glucose, it's O antigen. If all the same and is contain a galactose with a galactose and style is known as a antigen. It is found on the surface of the blood group A. If it contains galactose with galactose, so it is a B antigen found on the surface of the B blood cells. Okay, sphingolipid mutant to phosphocholine is stored. It's cause disease, Niemann-Pick disease. Galactose high if beta galactoside is mutant, so it's in GM1, so it's cause disease, genocide, gangliosides, and style galactoside is high if hexamide mutant, so it causes TSH disease. Glucose high if glucose cerebrosidase mutant, so it's cause a Gaucher disease. Okay, ceramide, glucose, galactose, and acetyl globoside. Stress if not released and acetyl by hexamelase AB, so it's cause disease, Sandhoff disease. Okay, if an acetyl is not releases by hexamelase A and B, so it's called disease, Sandhoff disease. It's ceramide, glucose, galactose. If galactose not released due to the mutant of alpha galactosidase A, so it's called Fabry disease. Okay, here's the mentioned structures, all that. Gangliomonocytes are the product during development stage of embryo called gangliomonocytes, in which ceramide attach or modify with glucose residue and galactose residue. Here ceramide, glucose, galactose, and acetyl galactose. And you know that if galactose high, because due to the mutant of beta galactoside is called disease, genocidal ganglioside. If acetyl galactose mutant, so it's called TSH disease. Glucose mutant, glucose high due to glucose cerebrosidase to a disease, Gaucher disease. Here's the structure in the form of the flow diagram. Now we talk about the cholesterol. Cholesterol is the essential component of the eukaryotic membrane, maintains self-fluidity of the membrane. Quite large structure, essential component of the raft, because raft is found in the case of the vertebrates. It's not found in the case of the invertebrate. It helps to dignity the cell structures and shape along with the sphingolipid. Normal phospholipids are structure cholesterol plus sphingolipid. Cholesterol have a 3-6 membrane fused ring with one pentagonal ring and 3 OH of the first 6 membrane outside membrane, so it's a polar. 3 OH of the cholesterol attached with the head of the sphingolipid, and structures look like that. It contains 27 carbon. Here's how cholesterol is derived metabolism. 7-dihydrocholesterol is the precursor of vitamin D. 2-step in skin UV rays ke through intermediate cholecalciferol vitamin D3. 1 step in liver and 1 step happen in the kidney. 125-dihydrocholesterol, 125-D3 vitamin A, mostly double bond in cis, but in the beta carotene double bond in the trans. Beta carotene equal no cleavage if cut middle half of 4,4. Retinol have 15 carbon chain in position 15th. OH undergo oxidize and form aldehyde group, so retinol become retinol. Okay, here's the structure, how this happened. Okay, so is enough for that. Now we discuss it. Biomembrane, can I cover it? Biomembrane, I think no. I should read first. Okay, bye. It quite helpful later on myself. Bye.